A sourdough starter is very easy to work with and one of the healthiest breads to make. As a start, you only need water, flour, and thyme to make one. The flour and water are mixed, and you feed them every 24 hours with more flour and water. After a few days, you should see it start to rise and fall. This is because of the metabolism of various microbes that grew from the flour, including lactobacilli, yeast, and other bacteria that are found in wine, vinegar, beer, and other foods that come from fermentation. Many types of microbes convert different sugars and molecules from flour into acids, carbon dioxide, and water. This acid is what contributes to the taste of sourdough bread, and the carbon dioxide waste is also what causes the starter to rise, as the newly formed gas is trapped in the dough. My personal big question was about comparing people's control groups to their experimental group in regards to how their cultures developed. Are all the strains the same in each of the cultures? Is the demographic of the habitat of the starter the same in regards to the dominant microbe species? Are there any microbes that are present in one but not the other? Each person in a group of over 150 had at least two starters, one control and one experimental. In the experimental groups, people put many kinds of fruit in them, although there was a limit of one fruit to each group. In the control group, there was just water mixed with flour. My personal experiments did not go so well, as both groups formed a hooch, which is a liquid consisting mostly of alcohol that forms because of conditions that are not ideal for microbial growth. I did not know that at the time, and my cultures mostly died a few days afterward. An easy way to fix the hooch problem is to feed the starter more often. To find the different species of microbes that were present in the starters, we had to use DNA sequencing for the entire culture. This included looking at a single gene in bacteria and a single region in the genome in fungi, the fungi being the yeast. There are two types of sequencing, shotgun sequencing and amplicon-based sequencing. Shotgun sequencing is looking at the entire metagenome, or all the genetic material of all the microbes, while next generation amplicon-based sequencing, the first thing to do is destroy the cells and remove the material except for the DNA. Then put the DNA in tubes and clone them using a polymerase chain reaction, where you use an enzyme to make copies of them over and over and over again. Then you create barcodes made of nucleotides that are unique to each tube. By looking at each region of a given unknown bacterium, you can identify its species and what tube it belongs to. This is how you get the species of the microbes in each sample of a starter. The original question I was going to ask was, does each smell correlate with a specific culture? However, this was difficult in answering because looking at the data of aromas versus orders of organisms of 2020's fall semester, many put their aromas down as not applicable. This could mean many things, such as there wasn't any smell, it smelled like flour, or the smell was indescribable. In addition, some microbes had a major presence in samples with a smell, but negligible presence in samples marked with not applicable. As a result, I refined my question to be, does everyone have experimental cultures similar to their control cultures? To find out, I studied two samples by Anna Nelson. She used blueberries in hers, and for the most part, the answer to my question in reference to hers was yes, the experimental group's demographics were similar to the control group. However, one major difference was that in the experimental group there was a relatively large population of Acidobacter, which had little to no presence in the control group. When researching Acidobacter, I found out that it is used to make beer, vinegar, and wine. It converts ethanol, which is an alcohol made by decomposing sugars, into acetic acid. Then the bacteria converts the acid to carbon dioxide and water. Converting the ethanol to an acid causes the experiment's pH to go down, and the bacteria has genes to protect itself from the acid, such as the production of citrate. With this in mind, I hypothesized that this bacterium would be found in already rotting sweet fruit due to the sugar content, and sour fruit due to the acid content. My hypothesis is directly tied to my refined question because it implies that the control groups have a distinct culture that does not have acetobacter in it, while cultures that have acidic or rotten sweet fruit in them will have that bacteria in them. However, my hypothesis was proven incorrect, at least with respect to sour fruit. With the 2021 spring semester content, I began studying the microbe cultures of one Han Parks, who created her groups with lemons, as our other group with raspberries was not included in my research. Looking through the results of testing the metagenome, there was a trace of Acetobacter, but it was minimal and definitely too low for me to see accurately in any of the groups. Although I had a sample size of just one, and so these cultures could just be a fluke, it could potentially open a way into disproving the acidic content of my hypothesis. Lemons are extremely acidic, but there was little to no trace of this bacteria in the starter. If my hypothesis is indeed incorrect, the blueberries that I originally found to have Acetobacter may have just come into contact with a source of that bacteria, or the bacteria may just be endemic to blueberries. In the future, I would like to test my hypothesis on other sour and/or acidic fruit to expand the sample size and get a better reading on if my hypothesis is correct or not. Then I would like to test my hypothesis on rotting sweet fruit like apples, pears, dates, and other vegetation with high levels of sugar. This will allow me to test whether my hypothesis is correct with respect to the sugar getting converted to ethanol by Acetobacter or another bacteria taking the place of that bacteria. Another thing I would like to do with this specific bacteria stemmed from a research paper I looked at when writing the transcript for this video. If I could catalog the genome of this microbe, I would like to find which genes control quorum sensing. Quorum sensing is when microbes sense other microbes in its vicinity and make decisions based on that information. For this bacteria, it would limit reproduction when the population is getting too high. This limits the fermentation process for many items, such as wine or vinegar, as bacteria in limited amounts can only do so much fermentation. Increasing the population by genetic modification to the quorum sensing would cause the fermentation to go much quicker.